Okay, let's dive right into this explainer. Look, if you're an MBBS student, you already know that microbiology can honestly feel like this endless, overwhelming ocean of facts. But today, we're throwing you a lifeline. We're giving you the ultimate high-yield cheat sheet for your exams. We're going to master culture media and inoculation methods together. We're breaking down exactly what you actually need to know, focusing on the acronyms, the visuals, and the concepts that your examiners absolutely love to test. So grab a notebook, get focused, and let's do this.
We're structuring this precisely the way your examiners think. First up, culture media basics. Then we'll tackle massive traps like enriched versus enrichment media. After that, we'll identify selective and differential plates, dive into transport and anaerobic setups, touch on modern automated systems, and wrap it all up with the key inoculation methods you'll need to ace your practicals.
So, Section 1, Culture Media Basics. Let's start with the absolute foundation here. What exactly are we feeding these microorganisms to actually get them to grow in the lab?
Well, to build a culture medium, you need a few core ingredients. You've got water and electrolytes, like sodium chloride. You add a little peptone, about 1%, which gives them partially digested proteins. Then throw in meat or yeast extracts, and maybe some blood or serum for those really picky, fastidious bacteria. But here is the absolutely crucial point for your exams: agar. Agar itself has zero nutritive value, literally none. Think of it purely as the concrete foundation of a house. It gives structure. It's a solidifying agent made from seaweed, acting like scaffolding for the bugs to sit on, but they definitely can't eat it.
And because agar is just that scaffolding, we can play with its thickness. Examiners absolutely love to test agar concentrations, so you need to lock these numbers in your memory right now. Your standard solid media, that uses a 1 to 2% agar concentration. But if you drop it down to 0.5%, you get a semi-solid medium. This is your go-to for demonstrating bacterial motility because it allows motile bugs to swim right through and make the whole thing hazy. On the flip side, if you crank it all the way up to 6%, it gets so stiff that it actually stops the swarming behavior of Proteus species dead in its tracks. Massive high-yield fact right there.
All right, let's look at the progression from simple to complex. On the left, you start with clear liquid peptone water or a basic yellow nutrient agar. But to grow those picky, fastidious bugs we talked about, you gotta upgrade. Add sheep's blood, and boom, you get blood agar. Now, if you take that blood agar and heat it to 70 degrees Celsius, the red blood cells lyse and release even more nutrients. That gives you this rich brown chocolate agar, which is the absolute favorite for fastidious bugs like Haemophilus influenzae that won't even grow on your standard blood agar.
Moving right along to Section 2, Enriched versus Enrichment Media. Guys, this is a massive trap for students. They sound practically identical, but functionally, they are entirely different beasts.
Honestly, you can literally copy this table directly onto your exam sheet. Enriched media, like those blood and chocolate agars we just saw, are solid plates. Their one job is just to provide a massive buffet of nutrients to grow fastidious, picky bacteria. Enrichment broth, however, is a liquid, and more importantly, it contains inhibitory agents. Its whole purpose is to isolate pathogens from a messy, mixed sample, like stool, by suppressing the normal flora so the actual pathogen can thrive. You'll want to memorize selenite F and tetrathionite for Shigella and Salmonella, and whatever you do, never forget APW, alkaline peptone water. That's the classic enrichment broth for Vibrio cholera. A super quick memory trick? Think of APW as always promotes watery diarrhea bugs. That'll link it straight to cholera for you.
Okay, Section 3, Selective and Differential Media. Building on that concept of isolation, how exactly do we pinpoint specific pathogens on a solid plate when we're dealing with heavily mixed samples, like stool or sputum?
That's where selective media comes in. These are solid plates explicitly laced with inhibitory substances, and you really need to be able to identify these on site. For example, if you see a stark, opaque green slope in a tube, that is Lowenstein-Jensen, or LJ medium, made specifically for isolating Mycobacterium tuberculosis. Compare that to a translucent deep green plate. That's TCB-S agar, used exclusively for pinpointing Vibrio species.
Let's lock in the acronyms for these selective media because, trust me, these are exam gold. We just mentioned LJ for TB and TCBS for Vibrio. Add these two to your list. Whenever you see DCA or XLD, I want you to immediately write down enteric pathogens like Salmonella and Shigella. Think extremely lethal diarrhea for XLD to remember it targets gut bugs. And PTA, potassium tellurite agar, that is your absolute trigger word for Corynebacterium diphtheria. Just think PTA, protect the airway for diphtheria. Easy, right?
Now let's talk about differential media, specifically looking at McConkey agar. Instead of inhibiting the bacteria, this plate actually color codes them based on their biochemistry. It's brilliant. If a bacterium ferments lactose, take E. coli for example, it produces acid and creates these beautiful, bright pink colonies. We call these LFs, or lactose fermenters. But if they don't ferment lactose, like Shigella, they just grow as these pale, colorless colonies, known as NLFs.
All right, shifting gears to Section 4, Transport and Anaerobic Media. What exactly happens when your lab is miles away, or when you're dealing with bugs that literally die in the presence of oxygen?
First up, transport media. The golden rule here is that bacteria do not multiply in transport media, they just stay viable. This stops delicate bugs from dying off and prevents the robust ones from totally taking over the sample. This table right here is a high-yield matching question just waiting to show up on your test. Lock in Carey Blair for your enteric bugs, like Vibrio, Shigella, and Salmonella. Just remember the name Carey means it helps you carry gut bugs safely. And for delicate Neisseria species, Amy's medium is always your top answer.
Now, what about strict anaerobes, like Clostridium, that absolutely hate oxygen? If you look at this tube filled with what looks like chopped meat particles, that's exactly what it is, Robertson's cooked meat, or RCM broth. It's the most widely used anaerobic medium out there. Why? Because those chopped beef heart particles at the bottom provide glutathione, which is this crucial reducing substance that gobbles up oxygen and creates the perfect low-oxygen environment. You can also use thioglycolate broth, which does a very similar job.
Moving on to Section 5, Automated Culture Systems. Let's leave the old-school manual methods behind for a second and look at the tech that's completely revolutionizing modern microbiology labs, especially for blood cultures.
When recovering a bug is really tough, like from blood or sterile body fluids, we rely heavily on automated systems. For your practicals, you have to know how they work. Bacti-alert systems are colorimetric. They detect the carbon dioxide produced by multiplying bacteria, which physically changes a sensor's color from blue-green to yellow. BacTech, on the other hand, is fluorometric. It uses a fluorescent dye that reacts when actively dividing bacteria start consuming the oxygen in the bottle. And if you're ever culturing TB automatically, remember the acronym MGIT, which relies on that exact same fluorometric principle.
Check out what these machines actually look like in practice. They continuously monitor the bottles every 10 minutes. And here's the brilliant part. These automated blood culture bottles contain special polymeric resin beads. So, if a patient is already on antibiotics, these beads literally adsorb and neutralize the antimicrobials floating around in the blood specimen. This allows the pathogens to grow way faster and gives you a much higher yield than if you just use conventional manual methods. It's a total game changer.
Finally, Section 6, Key Inoculation Methods. Let's wrap this up with some lightning-fast pointers on inoculation. You are definitely going to face these in your practical exams and OSCEs.
The absolute most common method is the streak culture. The prerequisite for identifying any pathogen is getting isolated individual colonies. To do this, you smear your primary inoculum, but the critical step, the one you cannot skip, is intermittent heating. You absolutely must flame your wire loop until it's red hot and then let it cool between each different set of streaks. This dilutes the bacterial load as you drag it across the plate, eventually forming this really nice feathery tail of individual colonies.
You can clearly see the result of that streak technique on the left, beautifully isolated individual colonies completely separated from the main mass. Now compare that to a lawn culture on the right. Here you're just swabbing or flooding the whole plate to create a uniform edge-to-edge carpet of bacteria. We don't use this to isolate anything. We use lawn cultures specifically for antimicrobial susceptibility testing, where we just drop antibiotic discs straight onto the lawn to see which drugs effectively kill the bugs.
Let's make sure your practical identification skills are rock solid. You need to be prepared to identify these two methods on site. When a liquid culture grows, you're going to see turbidity or cloudiness. A stroke culture is streaked in a zigzag fashion on an agar slant, which is perfect for biochemical tests. And finally, in a stab culture, the wire is literally stabbed straight down into a semi-solid agar butt. Like we talked about earlier with that 0.5% agar, this exact setup is used to test for bacterial motility.
So, let's put this all together. If an exited specimen, like pus from a wound, arrived in your lab right this second, would you streak it on Basal, Enriched, or Selective media first? Think about what we just covered regarding heavily mixed specimens versus delicate pathogens. If you can confidently answer that question, you've got the cognitive map you need to absolutely dominate your microbiology practicals. Until next time, keep studying smart.